Welcome. This is the 88th Cold Spring Harbor Symposium on Quantitative Biology, and I'm delighted to have Jeff Friedman here, who's been talking in the brain-body physiology. Jeff, we first met 30 years ago when you talked at Banbury about leptin. Maybe we should begin with a little summary of what leptin does and what's happened in that 30 years.
Well, I got older, but scientifically we identified the leptin gene as the target of a mutation that leads mice to become massively obese. Mice lacking the leptin gene weigh 3 times normal, have 5 times as much fat as a wild-type mouse, and eat voraciously. In '94, when we identified the gene, that was all a hypothesis, but in the successive years we've shown that to be the case. And much has been learned about the mechanisms by which leptin regulates appetite, largely by regulating the activity of key nerve cells, which is a topic of many of the talks of this meeting.
I was interested in one point you were making, think about neural circuitry that's involved, is that you started with Sherrington's very simple reflex, and then I guess then said that that could be a foundation, but then more complex behaviors get built on that simple foundation.
Yeah, I think my thinking on this topic has evolved. I mean, clearly when you initiate feeding behavior, it's to the exclusion of other competing behaviors, such as run away, drink, copulate, and so on. And so there has to be an elaborate switching mechanism that leads you to embark on one behavior to the exclusion of others, dependent on environmental factors as well as internal cues. And I used to think about that as a single part of the brain that acts almost like a switch. And I think that elements of that notion are true, but of course that single place where the decision is made has to get inputs, relevant inputs to generate a behavioral output. And thinking about how inputs are linked to output led me to the work of Sherrington, who was the first to map this precisely, first by describing reflexes, initially the scratch reflex of the dog. He looked at his dog who was scratching himself and said, or wonder what that's about, or if he knew that if he scratched him in a particular place, the dog's paw would shake, as we all know. And he looked at that simple situation and was the first to answer the question, what is this about?
So in relation to leptin and appetite, I guess, what are the other factors that the brain has to integrate, if you like, in terms of feeding or feeding satiated or?
So at a high level, 2 types of signals influence behaviors, including feeding. So-called interoceptive signals, these were described actually, or the term was first coined also by Sherrington. These are things like hormones and other factors that report your internal state. Leptin reports your internal state about how much fat you carry, but other hormones, which are the targets for new therapies for obesity, are hormones that signal other types of information, such as did you just eat, how much did you eat, how large a meal did you have, how much stretch was there in your stomach. These interoceptive cues are processed together with sensory cues, like is there a lion in proximity of the food I would like to eat? Or is the food rancid? Or just give a bitter taste which might indicate that it's poisonous? So all these internal and external cues are somehow processed to generate a behavioral decision. In this case, eat or don't eat.
So the leptin part of this is monitoring sort of fat content of the body, essentially?
Yes. So there are short and long-term systems that regulate appetite. The long-term system is one in which you try to maintain your fat stores at a constant level. And there's important evolutionary implications to this. If you have too much fat, that can be bad because you're more susceptible to a predator. If you have too little fat, you might be more likely to starve during the next famine. So biology has balanced those risks by creating a system that tries to keep your fat mass in a relatively narrow range. And that's part of a long-term system that monitors fat content. There's also a short-term system by which your recent food intake is sensed and conveyed to the brain. And these kinds of signals are largely gut hormones that react either to stomach or intestinal stretch or nutrient in the GI tract. These short-term signals in turn regulate both how hungry you feel between meals and how easily you get satiated during a meal. And these long-term and short-term systems interact to maintain your nutritional status at an optimal level.
Is there a single area of the brain that does this integration?
Well, most of these signals are read out either in relatively primitive areas of the brain, the brainstem and the hypothalamus. These regions are well conserved among all vertebrates, and they appear to respond to these interoceptive cues. The sensory cues I mentioned tend to be processed with ever increasing complexity in organisms by higher structures like your cortex and other brain regions that are designed to not just sense external stimuli, but to process them and make predictions of what those sensory signals are telling you. And as evolution proceeds, each organism is able to make more and more sophisticated predictions.
So you've now got a set of inputs. Are they integrated as though they were all equal, or do some take priority over others?
Well, there's a relativistic aspect to this, and let me return to my lion example for a moment, or predator example. Imagine you missed a meal and the only food around is in proximity to a lion. You would probably not make any effort to get that meal at all. If you were 3 days or 4 days into a famine, you might be thinking about a strategy such as throwing a rock or distracting the lion and running and grabbing the food if at that point starvation is a potential outcome. A more accessible example to us is, imagine you're 1 day into a fast and the only food available is rancid. You're not going to eat it. But 3 or 4 days of not eating, you're highly likely now to eat that rancid food. This was actually capsulized by Cervantes, who said that hunger is the best sauce. So all these stimuli are important, but they're contextualized based on your internal and external state.
You mentioned that these processes get integrated in, or assessed, if you like, in the brain stem and the hypothalamus. And you mentioned that this is a sort of conserved across vertebrates. For example, C. elegans, does it have, obviously not the same structures and organs and things, but do all organisms have some way of checking their food intake? And
yes, and it's actually quite similar, at least in the following sense. In the hypothalamus in particular, there are specific neural populations that drive appetite, and there are specific neural populations that turn off appetite. The balance of activity between these two pathways is regulated by these kinds of hormones or interoceptive signals. So the basic logic of the system is you have pathways that can push you in opposite directions, and then different stimuli can change that balance. That appears to be exactly the way C. elegans organizes their food intake. They have paired pathways that either drive or suppress appetite, the relative activity of which is balanced by internal signals. Now, of course, C. elegans don't have leptin or these other hormones, but they have other relevant factors that change that balance.
Yeah, so this is essentially a fundamental circuit that exists independent of particular ways that it's implemented.
I think that's right. I think the logic of many behaviors is that you'll have neural pathways that push you in either direction, and then internal and external cues can change the balance between those two.
Yeah, logic is the word. Logical circuitry is, I think, a good phrase. So we now know an awful lot more about control of appetite and our responses to appetite. Have these provided opportunities for therapies?
Well, leptin itself has very potent effects in a subset of the obese population, that subset that makes too little leptin. So some obese patients, as was the case for the OB mouse, make no leptin. And if they become massively obese, if you give them leptin, body weight and food intake go down. That also appears to be the case for the subset of the obese population that, for whatever reason, expresses constitutively low or lower levels of the hormone. But that's the minority. Most obese patients have high levels of leptin, and that appears to be a result of some insensitivity to the hormone as part of what's called a hormone resistance syndrome, that in this case is analogous to insulin resistance as a cause of type 2 diabetes. Most diabetics make much insulin. In the case of insulin, it's a very potent therapy if you're lacking insulin, but not so potent if you're insulin resistant. And the same appears to be the case for leptin. Leptin is not effective as a treatment, at least by itself, in most obese patients. The new therapies that have been developed then don't target the leptin system. They target this short-term system, the system that turns off appetite when you've overeaten. And they essentially mimic, in a very much more potent way, the signals that are normally generated when you eat a meal.
And this leptin resistance, is it mutations in the leptin receptor, or?
So there are genetic and acquired forms of leptin resistance. Leptin resistance is seen in patients who lack the leptin receptor or carry mutations in downstream mediators of that signaling pathway, largely what are called POMC neurons, which are the endogenous appetite suppressing neurons. And about 10 to 15% of morbid obesity is caused by one or a different single gene mutation in that pathway. This is, as I said, a minority of the patients, but it's a high degree of genetic loading. In most cases, the leptin resistance develops and the pathogenesis was unknown, although in a recent piece of work that is not yet published, we're able to show that in leptin-resistant mice, the cause of it is an acquired defect in the signaling of these same POMC neurons. So it's somewhat gratifying that, to the best of our ability to tell so far, the problems with leptin resistance map to the same basic circuit element.
Coming back to this integration, this inputs from various sources. So the obese mice are different, but in a patient who has a leptin deficiency, they can't, the input from other parts of sensory things like, I don't want to look fat, or I don't, are overridden by the physiology. I mean, these patients can't say, I don't want to look fat, I'm not going to eat. The physiology really drives them to.
Well, the bigger question here is what causes obesity, and there are three general factors. One is it's biologic. A second that some believe is that it's volitional, that the obese lack some level of willpower the lean exhibit. I find that lean people more often favor that point of view, and other people suggest that it's environmental. We live in a modern age where people no longer exercise, they have free access to food. I think the case of the leptin deficient children clearly indicates that biology is a major driver, but that's not to say volition and environment are unimportant, and if we were talking about any other trait, we would just immediately gravitate to the idea that there's a biological system on which environmental and volitional factors act. And I think that's the case here as well, that we have some basic biological circuitry, but that can be modulated by top-down controls, such as the conscious desire to eat less, or by environmental factors.
Well, it's almost lunchtime, so I'm going to let my volition go and get something to eat. Jeff, thank you very much.
Pleasure.